Good afternoon. It's my pleasure to welcome you to the Wednesday afternoon lecture, today specially designated as the Stettin Lecture. Before I introduce the speaker, Dr. Bonnie Bassler, let me say a brief word about Dr. Stettin, whose name is attached to this particular lecture. Dr. Stettin is someone who made enormous contributions to biomedical research and to NIH. He spent most of his career here looking over some of the things that he did in that time. One was that he was a recruiter, and I gather he recruited a number of very distinguished scientists, some of whom are still here, including Dr. Marshall Nierenberg, Nobel laureate, who was brought here by Stettin and obviously did pretty well in this environment and continues to. Dr. Stettin also, I gather, trained in his own lab as a postdoc, none other than James Weingarten, former director of NIH. So he must have had quite a magnetic effect on a lot of people. Dr. Stettin is perhaps best known here for his service as the third director of NIGMS, and there again led that particular institute with great distinction. So in recognition of those many contributions, he actually has two NIH entities named after him. This lecture is one of them, and the other is the DeWitt Stettin Museum of Medical Research, which was dedicated during NIH's centennial in 1987. Dr. Stettin died in 1990, but it's nice that we are able to recognize and remember him today as part of this lecture. It's my pleasure to introduce to you the speaker for the Stettin Lecture, a very distinguished speaker indeed, Dr. Bonnie Bassler of Princeton University, member of the National Academy of Sciences. Her work, which she's going to tell us about, will teach you, if it was not already apparent to you, that prokaryotic organisms are not necessarily the loners that you might think, but are actually quite social in their abilities to sense the neighbors around them, both of their own species and of other species. And the work that she and members of her team have done to identify the chemical signals that moderate those kinds of communications between bacteria both provide a wonderful window into how biology works and provide some interesting clues that might be utilized therapeutically in treating diseases like cholera. Dr. Bassler has originally received her training as a graduate student in biochemistry at Johns Hopkins, did her postdoctoral work at the Aguirre Institute in La Jolla, and has been on the faculty at Princeton since 1994. She's previously been a MacArthur Fellow, and as I mentioned, is a member of the National Academy of Sciences. It is a great pleasure and privilege to have her here to address us this afternoon, so please welcome Dr. Bonnie Bassler.
Thanks. He's trying to not infect me with a microbe. I'm absolutely delighted to be here. It's always a treat for me to get to come to this amazing place, the NIH, because of what you do for this nation, and also because I have a lot of friends here and fabulous scientists that have always been my heroes. So I'm glad to be back, and it's a huge, amazing honor. I want to thank Dr. Berg especially for inviting me to give this special lecture. It's a terrific treat for me. So you already got a hint of what I'm going to do, what I'm going to talk to you about. And so I really only have one goal in this seminar, and that's to try to convince you that bacteria can talk to each other. And if I manage that, a sort of secondary goal is to try to convince you that they're multilingual, and that also learning about the fact that bacteria can talk is going to, I think, going forward, have interesting medical implications for humans. And so that's it. So I've given my seminar, but now I'll give it to you in a little more detail. So I think that probably all of you know about what bacteria are. They're all single-celled organisms, and they each have one piece of DNA. So there's no internal compartments. They only have a few thousand genes, and they're the oldest living organisms on the earth. So they've been here for billions of years. And for all of those years, except the last about 20, they've been considered these asocial recluses. They live this kind of boring life where they grow to twice their size, they cut themselves in half, and one cell becomes two. And then it was always thought that each of these cells just goes out and does its own thing with no knowledge, if you will, of its sister cell. But of course, you hear about bacteria in the newspaper because they do all kinds of terrible things. What we don't hear as much about is all of the wonderful things that bacteria do. And I would first start this talk by trying to tell you that you have an amazing relationship with these critters that are these invisible, supposedly boring organisms. And so just to sort of set the stage for how I think about bacteria, I made this slide. This is my idea of a human being. So these are supposed to be all of the cells in the body of a human being. So about 1 trillion cells to make up a human and give you all of the features that you have. But it turns out that at any moment of your life, you have 10 times more bacterial cells than human cells in you or on you. So at best, you're only 90% human, 90% bacterial, 10% human. But then if you think about this in terms of DNA, because you guys are at the NIH, so you know it doesn't matter about the cells, it matters about your genetics. So here's my man, made up of A, T's, G's, and C's of his genome. And so of course we have the human genome. So there's 30,000 or so genes to give human beings all the things that they need to do and give us all our features and we do. It turns out if we look at the genetics of this, there's 100 times more bacterial genes in you or on you at any time. So no matter which of these metrics you like, you're maybe 10% human over here, and you're really more like 1% human if you think about the bacteria. And so these bacteria, we've known that these bacteria are there, but they've always been considered to be these passive riders, that they don't actually do anything for us, they're just free riders. And what's becoming incredibly clear now is that these bacteria play an active role in keeping us healthy. They're an invisible armor that keeps us from environmental insults. They make our vitamins. They help us digest our food. They educate our immune system. So what we're now becoming clear is that we live in this consortia with bacteria that are doing all of these wonderful jobs that keep us healthy, and we wouldn't be alive without them. And they hardly ever get pressed for that. What they do get pressed for is for when they make us sick. And so I don't want to be completely Pollyanna like up here. Of course, there are all these notorious bacteria that you hear about in the newspaper that have no business being in you or on you ever. And when they do get a toehold in you, they make you ill or they kill you. And so now that we know that bacteria have both of these sides, this good side and this bad side, the question from my lab was, given that these bacteria are so small, how could they do anything? Whether you want to think about them as the good guys or the bad guys, what we wondered is how could they ever have an impact on their environment? They're so small, how can they do anything to a big, enormous host like a human? And so the clue for how these bacteria actually get to do all of the things, good or bad, that they do, came from a crazy observation that Woody Hastings made almost 50 years ago. So he's a scientist at Harvard, and he used to like to hang out at Woods Hole in the summer, and he noticed that there were all these bioluminescent bacteria living in the ocean. And so he picked a favorite one, and you're looking at a picture of it. This is just a flask of a liquid culture of his favorite bacterium, which is called Vibrio fischeri. And so you're just looking at a person from my lab holding a flask of these cells. And what you can see is that the flask is glowing in the dark. And that's because Vibrio fischeri has the special property that it makes bioluminescence. So like firefly light, except the light is blue. And so to take this picture, all we did is turn the lights off in the room. This is what we see. We're not doing anything to the cells. They're simply making light. And so Woody Hastings noticed that, but he made a second observation about these cells, which is that they didn't always make light. What he noticed is when the cells were alone, they made no light. But when they grew to a certain number, all the cells turned on light together. And so what Woody wanted to know, and what we still want to know, is how can it be that a bacterium can distinguish times when it's alone and times when it's in a group and then do something? And what's fabulous about bioluminescence is that one can just see what the bacteria are doing as a group. And so we could use this and exploit this light to figure out how bacteria distinguish when they're in isolation versus when they're in a community. And so what we know is that in essence, the way it works is as follows. So this is supposed to be my bacterial cell. So when it's alone, this is Vibrio fischeri, it doesn't make light. But what it does do is to make and secrete small molecules that you can think of like pheromones or hormones. And we call these molecules autoinducers. And so those are supposed to be the red triangles on this slide. And when the bacteria is in a dilute situation, the autoinducers just diffuse away, and so the bacteria doesn't perceive them. But as the cells grow and divide in a particular environment, so it's not making light, but as they grow and divide in a particular environment, since all the bacteria are participating in making and releasing this molecule, the autoinducer, this autoinducer builds up in concentration in proportion to cell number. And so what happens is when the molecule hits a certain amount, the bacteria detect it, they turn on a signal transduction cascade, the output of which is bioluminescence. So what they're doing is counting one another by whether or not this small molecule is there. So in fact, they're talking to each other with this chemical word and telling each other how many of them there are. And so this is what Woody noticed almost 40 or 50 years ago now, and we understand now why Vibrio fischeri does this, why it turns on light at high cell number. And that has to do with the biology of the way that this bacterium lives. So Vibrio fischeri is this marine bioluminescent bacterium, but it lives as a symbiont in this animal. This is the Hawaiian bobtail squid. And so what you're looking at is a squid that's been turned on its back. And I hope what you can see are these two lobes that are glowing. These are a specialized light organ that's under the mantle or the body of the squid. And in this light organ, Vibrio fischeri lives in there in pure culture at something like 10 to the 11th or 10 to the 12th cells per mil. We have no idea how the squid grows the bacteria to this high cell number, but they're jam-packed in there. They're making and releasing that autoinducer, and so it's trapped in there in the light organ with the bacteria. It tells the bacteria, you're inside, not outside, you should make light. And so Vibrio fischeri glows. And in exchange for that, the squid feeds it. So this light organ is loaded with all these amino acids and sugars and goodies. It's a much richer, happy, fatter life to live in this light organ than to have to fend for yourself free living in the ocean. So the evolutionary selection from the bacterium's point of view is that it gets food and it gets to grow to much higher numbers than in the seawater. The reason the squid is willing to put up with these shenanigans is because it wants that light. And so the way that this symbiosis works, it's a very dramatic picture. The squid's only like this big, is that this little squid, this is called the afternoon, 3 p.m. lull. Anyway, this little squid is a nocturnal animal. So during the day, it buries itself in the sand and sleeps. But then at night, it has to come out to hunt. And so on bright nights, when there's lots of starlight or moonlight, since the squid is just in these shallow couple feet of water, that starlight or moonlight can penetrate the depth of the water the squid lives in. So what the squid has developed is a shutter. It's just the squid's ink sac that it can open and close over that light organ. And then the squid has detectors on its back, so it can sense how much starlight or moonlight is hitting its back. And then it opens and closes the shutter, so the amount of light coming out of the bottom, which is made by the bacteria, exactly matches how much light hits the squid's back. So the squid doesn't make a shadow. So it's actually using the light from the bacteria to counter-illuminate itself in this anti-predation device so that predators that would see its shadow, calculate its trajectory, and eat it can't find it. So this is like the stealth bomber of the ocean. It makes itself invisible to predators, and that's why it's willing to feed this enormous culture of bacteria. But then if you think about it, the squid has this terrible problem because it's got this dying 10 to the 12th cells per mil culture of bacteria, and it can't maintain that. So what happens is every morning when the squid buries itself in the sand to go to sleep for the day, it's got a pump that's attached to its circadian rhythm. So when the sun comes up, the squid buries itself in the sand, but it pumps out like 95% of the bacteria. So now the bacteria are dilute, and that little hormone molecule, the autoinducer, is gone, so the bacteria aren't making light, but of course the squid doesn't care because it's asleep in the sand. And as the day goes by, the bacteria grow and double, the molecule builds up, and at night the light comes on exactly when the squid needs it. So it's like a squid chemostat that lasts the lifetime of the squid. And so the reason to tell you that story, besides the obvious one, is because this is the first one of these bacterial systems that we learned about this idea of chemical communication from Woody's work. But what's become clear in the last 10 or 15 years is that probably, and this is a big word, all bacteria have chemical communication. What we now understand is that this is not some anomaly of this ridiculous bacterium that lives in the squid. We now have hundreds and hundreds of examples of bacteria, clinically relevant bacteria in many cases, that have a program of gene expression that they carry out when they're alone. And among those genes are a gene that's a synthase that makes an enzyme that makes a molecule that they release into the environment. So at low cell density, those molecules float away, and the bacteria carry out behaviors that are selected for being an individual. But then when the bacteria grow to high numbers, these molecules accumulate, and they switch on a gene expression pattern that's really good for acting as groups. And so now we have a fancy name for this. We call it quorum sensing. The bacteria vote with these chemical votes. The vote gets counted, and then everybody goes along with the vote. And so there are hundreds and hundreds of examples of these things now, and typically in each of these regulons, there's several hundred genes that are controlled by quorum sensing. So when a bacterium makes the decision from going it alone to acting as part of a community, it's a big deal. It's like development in an embryo, and they have to turn on this timed program of gene expression that is a big part of their genomic personality. So that's how it works. We also now know that this is not just about bioluminescence. And so again, I've told you that there's hundreds of examples. I just put a few bacteria on this slide to give you examples of some of the quorum sensing controlled behaviors that we know about. So I told you about bioluminescence in Vibrio fischeri. It's only good to do that when the cells are all together, or else you don't get visible light. The thing that people have been most interested in the last decade are virulence factor expression and biofilm formation. So biofilms are how bacteria live. We now understand they live in these films in us or on us attached to surfaces. These are big communities of bacteria. You need lots of cells working together to make the biofilm. And then also the secretion of virulence factors. And so very often these kinds of traits are controlled by quorum sensing. And so the idea is if a tiny little bacterium that's not supposed to be there gets in you, the worst thing it can do is to just start secreting all of these virulence factors, because there aren't enough bacteria to make any damage. And furthermore, these virulence factors, these exoproducts, tend to be huge red flags for the immune system. So the better idea is the bacteria get in, they wait. They count themselves with these small molecules. They recognize when they have the right number that if they all launch this attack in synchrony, they're going to be able to overcome an enormous host. So whether it's a symbiotic or a hostile relationship, what we now understand is that quorum sensing controls all kinds of things that bacteria are going to give away, and you never have a prayer of getting your own protein back. You're depending on the community to participate in the behavior, and then the collective flourishes. And so that's what we understand now about quorum sensing, and that probably all bacteria have these kinds of systems that allow them to act in groups and do things that they could never accomplish if they simply acted as individuals. So that's what they do. We also now know about the chemistry in many cases that are involved. And so again, this is just a potpourri of different bacteria that we have studied their quorum sensing systems. In the case of gram-negative bacteria, what we know is that the word that they talk with are what are called homoserine lactone molecules. So again, just a few bacteria, and this is the molecule that each one of these uses as the word. So those were the red triangles on the slides before. And I hope what you can see, of course, is that all of the molecules are related. So they all have this lactone head group, but then what you can see, these are just carbons, is that these little acyl chains are slightly different in every single species of bacteria. And what that does is to confer exquisite species specificities to each of these languages. So each of these autoinducers interacts with its cognate receptor protein and no other. So these molecules allow intra-species communication. This is how bacteria count their siblings. And so what I mean by that is if I take the Vibrio fischeri molecule and put it on Serratia, nothing happens. Likewise, the Serratia molecule has no influence on Vibrio fischeri. So gram-negatives talk with homoserine lactones. We also now know that gram-positive bacteria have quorum sensing, and so the idea is exactly the same. They're recognizing the buildup of these extracellular molecules, but they use peptides as their autoinducers. And so again, I just put a few of the peptides that we know from different species of gram-positive bacteria. Sometimes they can just be the naked peptide, sometimes they can have a bell or a whistle, a modification on one of the amino acids, or they can be cyclized. But the rules are exactly the same. Even with these 4 peptides that are only different by one amino acid, they are exquisitely specific for the bacterium that made them. So again, peptides are the language of gram-positive bacteria, but they allow them, gram-positives, to talk within their own species. So that's what we learned first, that there seem to be these 2 big kinds of molecules that, for reasons that we don't understand, are restricted to the gram-negatives and the gram-positive bacteria. But then, if you go back to the beginning of my talk, of course, what we're now starting to learn about is that bacteria don't live in isolation. They live in these amazing communities, like on our body, of hundreds or thousands of different species, living together in an environment. So this, for example, is a picture, a micrograph taken from your elbow. And so this is just the bacteria that are on human skin at any time. And of course, you can see that each of these different shaped bacteria is a different species. And so the question that we had after we figured out about the homoserine lactones and the peptides is if it's really true that bacteria are trying to count with these molecules, it can't be enough to only be able to talk within your own species. Because if you're really trying to take a census, there must be a mechanism for understanding whether there are other species in the community. And so by doing genetic and chemical screens, indeed, we found a molecule that appears to be a universal molecule that is made by both gram-negatives and gram-positives. So we call this molecule autoinducer 2 because it was the second type of molecule we found. It is made by an enzyme called LuxS, which is in the genomes of both gram-negatives and gram-positives. And what we showed is that LuxS does one thing. It makes this molecule. And this molecule is used for cross-species communication. Right, so all the bacteria make and respond to this molecule, and so it doesn't have the species specificity of the homoserine lactones or the peptides. We call it autoinducer 2 because the name of it is 4,5-dihydroxy-2,3-pentane-dione. But anyway, that's why it's called autoinducer 2, and I'll refer to it that way going forward. So what we're now understanding at this point in the game is that bacteria must have a minimum of 2 languages. So they have to be multilingual, in that they have to have some molecule, a peptide or a homoserine lactone that says me. And then they use autoinducer 2 to say other. And so what we think the circuitry looks like in these different bacteria is actually depicted in this cartoon. We now know that bacteria have an enzyme or a gene that encodes a species-specific molecule, so a homoserine lactone or a peptide that gets detected by a partner receptor. And then they also have this enzyme LuxS that makes autoinducer 2 that's also released into the environment, and then they have a cognate receptor that is specific for that molecule. And then this information, as these molecules build up, gets transduced into the cells to control these hundreds of genes that underpin group behaviors. And so what we're starting to realize is that bacteria can distinguish self from other using these 2 molecules, and what they really can do is to measure blends of these. And so they can ask, if I can say those things, questions about how many of me and how many of you are out there. And then the genes that get turned on and the order that they get turned on in has to do with whether there's more of me or more of you out there. They carry out different tasks based on whether they're in the minority or they're in the majority of any given consortium. The molecules that we have not found yet are the ones that say who the other guy is. We know those molecules exist because you can't get these beautiful bacterial architected communities without knowing who your neighbors are, and that's what people are hunting for right now in my field. So there has to be at least one more class of molecules out there. And so that's my extended introduction as ordered. And so now I can't resist. I want to tell you a brand new story from my lab about trying to do something practical. Now that we've learned in the last decade or so that these bacteria talk to each other, we know they carry out all these group behaviors, and we know that a big part of that is going to be virulence and biofilm formation. And so the question is whether or not we can manipulate these quorum sensing systems to make new kinds of therapeutics that would no longer kill bacteria, but would just make them so they can't talk or they can't hear, and maybe these could be used as new kinds of antibiotics. And so for the rest of my talk, I'll tell you our first steps in trying to do that. Okay, so this is the same cartoon I showed you a minute ago, and so the strategies for doing this are actually, obvious. One is if you wanted a species-specific drug, you want a Pseudomonas drug, what you would do then is to try to either make a bacterium to antagonize either signal production or signal reception in the intra-species communication system. The alternative is that we also need, there's a place to have broad-spectrum antibiotics, and so the possibility exists, since this autoinducer 2 molecule is interspecies and seems to be generic, maybe you could make strategy to interfere with production or reception of autoinducer 2, and that might be able to be used maybe in both gram-negatives and gram-positive bacteria. And so we have been working on both of those strategies, and the last time I was here I talked about this one. So today what I'll do is tell you about our efforts to try to shut down intra-species communication with one of these homoserine lactone autoinducers. So we're trying to target one of these systems. Okay, so now I've told you about the molecules and what we're doing, but I have to tell you how bacteria actually detect these molecules. So what we know is that there are two mechanisms for detection of homoserine lactones. So the first one exists in all of the Vibrios. In that case, they have a synthase that makes a particular homoserine lactone that is delivered to the outside of the cell, and then on the cell membrane they have a 9 transmembrane spanning protein which is called LuxN that detects the autoinducer in the periplasm. So the autoinducer gets out, the LuxN protein is sitting there like a little antenna, and it detects the autoinducer out here. And the autoinducer binding event initiates a phosphorylation cascade, so phosphate flows through a signal transduction cascade inside of the cell, a transcription factor gets phosphorylated, and all of these group behaviors turn on. So the autoinducer gets detected on the outside, and information is by a phosphorylation cascade. So this is how all the Vibrios detect homoserine lactones. All other gram-negative bacteria that we know about, and so there's hundreds of examples of this, have a different circuit for detecting homoserine lactones. What they do is they synthesize their homoserine lactone autoinducer, it diffuses outside, and then it diffuses back into the cell. And it gets bound by a cytoplasmic protein that is called LuxR, or the LuxR type proteins. So these proteins are soluble cytoplasmic proteins. They bind the autoinducer, and when they're bound to the molecule, they sit on DNA and they turn on the quorum sensing genes. So these LuxR proteins are transcription factors. Okay, so the autoinducers are similar, but the method of detection is different. One's a phosphorylation cascade, one's a transcription factor, and where the autoinducers get detected are also different. One set gets detected on the outside, and the other gets detected on the inside. And so we wanted to see if we could inhibit any of these systems. And so we started over here with this system with the Vibrios, because I work on these bioluminescent Vibrios. And so what we wanted to do is to start on a free-living bacterium, Vibrio harvii, that's related to the one that I told you about Woody Hastings. And then the reason is that I've already given a plug for is because the readout of quorum sensing is so easy to measure. It's bioluminescence. So the bacteria naturally make bioluminescence in response to this molecule. If we could shut that down, if we could make antagonists, then we can simply look for inhibition of quorum sensing by reductions in bioluminescence, and you can measure this very quantitatively over 6 orders of magnitude. So what we did then, and this was a postdoc in the lab, Lee Swem, what he did was he started with our Vibrio harvii reporter strain. This is the autoinducer, the native signal, that when it binds to LuxN, bioluminescence turns on. And then what he did is he just took this strain to the Broad Institute in Massachusetts, and we screened their library for molecules that shut down bioluminescence. And so sure enough, he got those molecules just by doing a random screen, and then we came back and made sure that the molecules work by shutting down LuxN, not by doing something awful to the cell. And so he got about 15 molecules that he liked out of that Broad screen. And what I did here was to simply put a few of the structures that came out of the screen. And so what I think you can see is that these molecules look nothing like the native signals. So these aren't molecules that one would make if one were trying to do this by rational design. So he got these different molecules, and then he brought his most potent one. He went ahead and studied this and how it works on LuxN. And what he showed is that this molecule is a competitive inhibitor. And so what that means is that it slots into the same binding site in that 9 transmembrane region that the autoinducer fits in. So he could show that they're competing for the same binding site, and by doing mutageneses and suppressor analyses, he could find the amino acids in this big transmembrane domain that are important for binding the autoinducer and for recognizing the antagonist. And so we could find those, but the problem, of course, is that this protein has 9 transmembrane domains. So trying to get a crystal structure or a structure of that interaction, either with the ligand or with the antagonist, we can't do it. But we could get these inhibitors that we knew were competitive. And so what Lee thought then was, now, wait a minute, he knew about these 2 ways that bacteria have to detect autoinducers. So he went to the Broad and he got the antagonist on Vibrio harvii, but he wondered if even though this receptor and these cytoplasmic LuxR receptors, even though they're not evolutionarily related, they don't sit in the same compartment of the cells and they don't function by the same mechanisms, both of them have to fold up to be able to bind a homoserine lactone. So he wondered, could he take the molecules that he got in this screen and get them to antagonize in this other context, because that binding site must somehow look the same because it accommodates in the wild a similar molecule. So what he wanted to do was to get a system up to look at this kind of quorum sensing. And so what he did was he chose a bacterium to look at, which is called chromoviolaceum. So it has an autoinducer synthase that makes this molecule, which has a 6-carbon acyl chain, and it gets detected in the cytoplasm by a protein called CVIR. And then again, when it binds that 6-carbon molecule, it sits on DNA and turns on the quorum sensing genes. And so the reason that Lee chose this organism, there's actually sort of 3 reasons. The first reason is because this is a terrible pathogen. And remember, we really would like to move this out of the realm of the bioluminescent bacteria and into thinking about therapeutics. So he wanted to try to antagonize quorum sensing in a real pathogen. The second reason is because the molecule, the C6 autoinducer, is pretty close in size to the Vibrio harvii one. So this is a C4, and remember the antagonist worked on this receptor, so he thought there would be a good chance of this working because the native ligands look kind of similar. And then the third reason he picked chromoviolaceum is because one of the quorum sensing readouts, one of the genes that's controlled by quorum sensing, encodes the enzymes that make a beautiful purple pigment, which is why this bacterium is called violaceum. And so chromoviolaceum is purple, the wild type, and if you knock out either the autoinducer receptor or the autoinducer synthase, the bacterium is white, because the secondary metabolite, which happens to be purple, is one of the hundreds of quorum sensing controlled genes. And so if you don't have bioluminescence, the next best thing is purpleness. And so what he thought was that he could do his screens and look for reductions in purple the same way he had done for Vibrio harvii using bioluminescence. So that's the reasons that he chose that strain. And so remember, he had these 15 molecules that inhibited quorum sensing in Vibrio harvii. And then he wanted to test, do any of them inhibit production of violaceum? And so that's shown on this slide. We can just extract that purple pigment and read its absorbance. So this is purple on this axis. And so what he did was he made a mutant that doesn't make its own homoserine lactone. So there's no signal there. So of course, this makes no purple pigment. If we add back exogenously the C6 homoserine lactone, violaceum production turns on. That's just quorum sensing. And then one by one, he tried his different molecules. And sure enough, some of these molecules inhibited. And so what that means is that the ones that inhibit actually get into the cell, right, and bind that CVIR receptor and shut it down. And the one that he liked the most I have labeled in red because it completely inhibited quorum sensing. And so now I'll show you that molecule. And so this, I just put a comparison. This is the native signal, the autoinducer for chromoviolaceum. And this is the one of the 15 molecules that work the best from the Broad collection that Lee brought back. And so now what I hope you can see is that this molecule really is an analog of the autoinducer, right? So this part actually does look a lot like the native ligand. And so it was kind of obvious to us why this should be an inhibitor. It looks like the signal. So now we have molecules that inhibit externally LuxN and they inhibit internally CVIR. And what we want, but they're just these molecules that came out of this library of random molecules. And so what we wondered is, could we make these molecules more potent? And so we started with this one as our favorite molecule, and we made a focus library of about 100 different molecules based on this core structure. And so I just put a few of the bells and whistles that we hung off the end of that different molecule. And then again, using this violaceum purple assay we just tested, are any of them better than the original molecule? And so of course, most of them were duds, but we got a couple that actually were more potent. And so here they are. So this is the original molecule that Lee got out of the screen. And so its inhibition constant is 2 micromolar. If we take this methyl group off, in fact, that makes the molecule twice as strong. If we do that and we actually take the sulfur out, so now we really make this look like the homoserine lactone, we give it a homoserine lactone head, then what you can see is that this molecule is now 5 times stronger than the original molecule. So by tinkering with the molecule using chemistry, indeed, we can find sites on the molecule that we can change to make these molecules better as antagonists of quorum sensing. And then what Lee went on to do is to ask, how do these work in the chromoviolation strain? And what he showed is that the protein folds up perfectly fine around these molecules, but somehow these molecules lock the protein into a form that is unable to bind DNA. So the protein is soluble, the antagonist is bound, but it simply can't bind DNA, and that's why these are inhibitors. They can't bind DNA, you can't turn on the quorum sensing response. So he did a number of experiments that I won't show you to prove that that was the case. So now what Lee did was he originally got molecules here, he tried them here, he made them better here, right? So now they're stronger. And so the question is, are the features that we introduced into our antagonists that make them better inhibitors of CBIR, will those be conserved over here? And so then what he did was he took the more potent molecules and tried them against Vibrio harvii, and sure enough, they got stronger. And so what that means is that even though these two receptors have nothing in common at the amino acid level or at the mechanism level, the binding site, which accommodates a homoserine lactone, must have these features conserved, that when we make the molecules better antagonists in this context, it also works here. And so now we would really like to know, what is it that we are doing to those molecules that make them stronger antagonists in both of these setups? And so what was nice about the second setup, I've already alluded to, is that this protein, this autoinducer binding receptor, is a soluble protein that lives in the cytoplasm of the cell, so we could purify it and crystallize it and solve the structure, both with the ligand bound, the native autoinducer, and also with our antagonists. And so Lee did that. And here's a structure of the CVIR protein, and this is with the autoinducer bound. So this is the native ligand, that the C6 molecule, that when it's bound here, this protein binds DNA and turns on 100 quorum sensing genes. So he also crystallized it with one of our antagonists, and so what I've done now is to put the crystal structure of the antagonized form right on top of the form with the homoserine lactone. So the green protein with the red molecule, that's the native ligand. The blue protein with the longer, bigger molecule, that's the protein that's got the antagonist in it. And I hope what you can see is that these proteins look identical. So there's like almost no change in the protein. If we looked over the entire protein, there was nothing that was different than by more than 1 or 2 angstroms. And so the problem with that is that we know biologically these proteins are doing very different things. One is an active DNA binding protein, and the other one can't bind DNA. So even though when we looked at the crystals originally, they look basically the same, there had to be something different. And so when Lee looked really closely, what he noticed is that there's a methionine that sits at the bottom of the ligand binding site in the native protein. So this methionine sits like a lid holding the autoinducer into this ligand binding site. And when he looked at the crystal structure with the antagonist bound, what he noticed is when you sort of jam this bigger molecule in there, it kind of elbows this methionine out. So this methionine, now you have to look at the blue one, changes by 1.5 angstroms. And that was the only obvious difference that we could see in the protein. And so what Lee wondered is it enough to simply tickle that methionine residue, just move it out of the way a little bit, is that enough to change a molecule and a protein from an agonized form to an antagonized form? And so what he thought he could do to test that idea is to simply put a smaller amino acid here. If he put a small amino acid there, then this long extended molecule should fit in, and maybe he could change an antagonist to an agonist. And so what he did then was he simply replaced this methionine with serine and asks, how does this work? Obviously, I wouldn't tell you this if it didn't work. So now, you're looking at violation production, right? So with wild-type CVIR, with the native ligand, you see that the purple quorum sensing turns on. But the big molecule, the antagonist, there's no quorum sensing. If Lee simply changes that methionine that's at position 89 to serine, so he just makes a little extra room, now the antagonist is actually a perfectly fine agonist. And we've also done the reverse. We can change an antagonist or an agonist to an antagonist by simply changing 2 amino acids in the binding site. And so now where I think we are in this project is that we can get these molecules, and then we can know by using chemistry what features of the molecules we need to change to make them good agonists and good antagonists. And then we can also simply look at these proteins. We don't even need anymore all of the crystal structures. We can just thread these proteins through programs and know whether a particular molecule is going to be an agonist or an antagonist by what amino acids we know are sitting in the binding sites. And so now we're able, in a much more sort of systematic way, to be able to do chemistry and know what the outcome of a particular change in a molecule is going to be. And we can also do mutageneses of these proteins and know how the mutants are going to behave toward the molecules. And so I think we're getting to the step that's much past just going to libraries and screening through hundreds of thousands of molecules. We can actually make these pairwise combinations and know whether they're going to be active or inactive as agonists and antagonists. And so that's how far we've gotten with this, and we've done this in a number of other contexts, and so it's starting to work, that we can just look at these proteins and these molecules and know how they're going to interact. But of course, I sort of alluded to the idea that, in fact, maybe we should be able to make therapeutics out of this. And so even though we don't have the perfect antagonists yet, these are just lead molecules that have been refined a little bit, they do not have drug-like properties yet, we do have what seem to be potent antagonists of quorum sensing. And so this is the one that I've been talking about the most. This is our most potent molecule as an antagonist. And so the question is, yes, we can shut down quorum sensing in these bacteria. We know we can do that now. We know why the molecules work. But is there really any merit into thinking about this in terms of a strategy for a new kind of antibiotic? So before we go on and make lots more molecules, maybe we ought to just test whether or not this works. And so what Lee wanted to do was to move this into an animal model and see if he could shut down quorum sensing in an animal and improve the outcome of the host. And so what he decided to do for the first experiment is to simply use C. elegans. And so you guys know C. elegans is this wonderful model system for development in neurobiology, but it's also turning out to be a fabulous model system for pathogenesis. Because what worms do is that they eat bacteria for a living, and so the way people grow them is they simply put the worms on a puddle of bacteria and they eat them, and that's how they stay alive. But if you put them on a puddle of a pathogenic bacteria, the worms have to eat them, but then the worms get an infection and they get killed. And so we can actually use that to see whether or not we can use our anti-quorum sensing molecule to fix this worm. So Lee went ahead and showed that chromoviolaceum is a pathogen on this worm, and then he showed that chromoviolaceum needs quorum sensing to kill the worm. And so that's shown on this slide. So this is just a lifespan assay of the worm. So worms typically live 2 weeks, so this is days, and this is the percent survival. So if we simply put the nematode on chromoviolaceum, that's the red lines, what you can see is that the chromoviolaceum causes infection and all the worms die in a couple of days. But if Lee either knocks out the quorum sensing receptor or he knocks out the synthase, so the bacteria can't hear or they can't talk, they can't do quorum sensing, what you see is that the worm lives perfectly fine. And so we don't actually know what the toxin is that chromoviolaceum makes that kills the worm, but we know it's under quorum sensing control. So you have to have quorum sensing to kill the worm. And so now the question is, can we put a wild type bacterium down there with the worm, give it our molecule, and get the worm to live longer? And the answer is yes. And so here's the final experiment that I'll show you. So this is percent survival over time. So if we put the nematodes on wild type chromoviolaceum, they all die in a couple of days. If we simply add about, this is 20 micromolar of this chlorolactone, this potent antagonist, what you can see is that we can greatly extend the lifetime of the worm. So we think that this we're happy about this result because we think it shows that it's actually worth going forward and thinking about these things. And of course, what we really need to do is to use an animal that we can treat over time that's a mammal, right? And so what Lee has now done is to move this into mice, and so we have a mouse system up and going. And then, of course, we're trying to see how far we can push this with how many pathogens. And so we have lots of nice mouse models for different, much more clinically relevant pathogens that Lee is testing all of these inhibitors on. And so that's where we are right now, and I hope what you think, is that bacteria can talk to each other, and the way that they talk to each other is with chemical words. That what we think that this means is that bacteria are multicellular. So by being able to do these collective activities, by talking to each other, by synchronizing gene expression, we think that this is where multicellularity came from on this earth. We think bacteria invented the rules for that, and that hopefully by studying these collective behaviors in bacteria, these simple systems, beyond making these therapeutics, we hope that we're going to be able to gain insight into how collective behaviors work in higher organisms as well. And then I didn't talk too much about the interspecies communication, but what we now know is that what bacteria also invented is the ability to distinguish self from other. So by having a molecule that says me and a molecule that says other, we know that the bacteria are able to do that. And we would argue that, of course, that's also what happens in your body. It's not like your kidney cells get all mixed up with your heart cells, and that's because there's all this chemistry going on that tells different groups of cells who they are and what they ought to be doing. And again, we think that some of the rules for how those processes work happened in bacteria a long time ago. And then, of course, I think the major thing that's happening in my field is this idea of trying to stop quorum sensing, so making both industrial and biotechnological products that could impede quorum sensing in pathogens. But just to give a plug one more time for all of the bacteria that are really beneficial and that we can't live without, the other thing that's happening in my field is this idea, if we could make pro-quorum sensing molecules, so if we could make molecules that made our commensals able to talk better or better at collective behaviors at the expense of these invaders, maybe this is the place where we can move probiotics from, sort of the fringy, health food store, but into the realm of rigorous science, and that maybe that actually is the best strategy, is to simply beef up the conversation of the commensals. And so what I didn't tell you about today is that we've also been collecting up all the agonists we can find, trying to mess around with this in the opposite direction, like make these bacteria better at quorum sensing. And then the final thing to end the talk is to make the confession that we didn't think any of this up. So now that my field has realized that these bacteria are talking to each other, and then of course lots of scientists are now interested in impeding quorum sensing, people in the field have started to think, well, maybe the bacteria already thought this up. And so sure enough, what we now understand is that if you look out in sort of natural populations, you can find bacteria that eat each other's autoinducers, that eavesdrop, that free ride. Like there's guys out there that talk with the homoserine lactone, and the guy next to it in the dirt makes a lactinase, so this poor bacterium is trying to talk, and the guy next to it is making it mute. They make all kinds of antagonists, they do all this cheating and misinformation. And so maybe, in fact, instead of trying to do this the way we're doing it, we should just be looking out in these natural populations, and the best strategy, which has been tested over billions of years, are actually out there, and we should just be discovering them. And so I think that's a big thing that's that is happening in the field now, is just to look at what's out there in the bacterial world, and there's some beautiful sophisticated strategies for doing this chemical warfare among the bacteria. And then finally, this is Lee and Danielle Swem. So they did all the new work that I told you about. Lee's a postdoc and Danielle is a technician. You might notice they have the same last name. And today I gave you that's because they do. And today I gave you a chemistry talk, but what I should say is that we are mostly geneticists in my lab, and this is the best experiment that they did while they were with me. So those are, we give those kids some toothpicks and get them going. And then, so they did all the new work on the antagonists that I told you about, and then this is the gang that did all the work to discover autoinducer 2 and the interspecies communication and the older work. And so again, thank you tremendously for having me here. It's a big treat.
It's very actually exciting story about this words, that if I can use this word.
I do.
So my question is somehow related to a little bit more to your first part of your talk. So if we speak about languages, and maybe we can go back and use information theory to look at this. So what would be your estimates in general, how much information is going in between bacteria and different species and bacteria is the same species? And actually, another thing that you don't talk at all, how much information we exchange between host and different bacteria?
Yeah, so that's where this is going. So we work with Ned Wingreen. He's a theoretical physicist. He does information theory, and we actually think about those questions a lot. What I can tell you is that they get more than 2 bits of information by making these blends, but always, big word, in the systems we've studied so far, my signal is stronger than yours. So my vote counts more than yours. So always the intra-species signal is stronger than the interspecies signal. So they definitely care or pay more attention to their own species than they do to the others. That we know. And then in terms of like whether they are tuned into or receiving signals from the eukaryotes, it has to be like that, right? And I think that's the place. So people are starting now to try to look at what information, other than touch, are they getting, what chemical information are they getting from the host? And we don't know, anything about that yet, but there's lots of people working on that as well, like whether we're either eavesdropping or actually participating, like this is a dialogue between the bacteria and the host. And I think it's going to depend on whether these are hostile or symbiotic relationships, but I think that's not very far away before people know some of those molecules.
And the second question related to people working in Quorum Sensing says that it's also related to how fast different species move and how distant they are from each other. So would you comment a little bit?
Absolutely. So this is about being in the vicinity, right? But of course, that's what, if I'm far away, I should be acting as an individual. And so I only showed you, we actually know 4 molecules, so I only showed you 2 kinds today. So for sure, this is all about your location. Like when Vibrio harvii is swimming around in the ocean, it's not, by itself, it should be in the program for single cells, right? It's only when they're on a surface, when they're, in an organ, when they're in an animal, and they're dense, that quorum sensing works. And what we're also starting to learn about the molecules is that some are very short-lived, some are very long-lived, some are incredibly hydrophobic, some are incredibly hydrophilic. So there's all this spatial and temporal information, besides just number, encoded in those signals, right? So I think it's a very sophisticated computation that they're doing. It's more than self and other and number, right? It's also how far away, how greasy is it, how hydrophilic is it, right? And how long do these signals last? And that, I think they are accessing all of that information by making these different chemicals.
Nice talk. And I was wondering, you sort of alluded to this at the end. We're going to talk about complexity here, where there is really one organism enmeshed in a sea of other organisms. And how do you propose to really think about this type of complexity? And that's certainly going to be what you're going to go as you move from C. elegans to mice, is the interaction of this chemical on the entire microbial community.
So if you're asking about the therapeutic or about
even
just
the
in general,
quantitative assessment of it.
Right. So taking this a step up to, you have mixtures of bacteria, right, is like, it's the level that my field has not been able to deal with yet, right? We're getting there, right? I think in defense of my field, right, we had to learn some of these things first. And now I think people want to put these bacteria in mixes. We're certainly not going to do all 600 species in your teeth. We're going to do 2, right? So that's what people are doing, are trying to think up simplified mixtures of species which would see each other and which have a good likelihood of interacting, because I think that's the only way we can find that next set of molecules, right? They don't make them if they're by themselves, right? So I think that the ticket going forward is going to be this 2 or 3 species consortia, right? And then, and you're absolutely right. Like everything we've done, the bacteria are by themselves, right? We dump these molecules on, right? And so we have to start thinking of them in a more complicated way, and I think that is the future of this field. In terms of like the therapeutic, you are going to affect all the microbes, but that's what every antibiotic has always done. It's, you know what I mean? And so like what's so remarkable is that the last 60 years the antibiotics actually worked.
Oh, I know.
Right? And so so we're banking on that history that where you killed everybody and your own microflora comes back, because you have these persisters or you eat a lot of it, right? And what we're doing is a much softer selection, but we get that all the bacteria are going to be goofy if we make this anti-quorum sensing molecule.
And I guess there, it's really the analogy you talk about words. Is there going to be a grammar to this language?
Yeah, and
is there going to be a finite, fixed vocabulary?
Right. So I think, you know, like you can argue like these Vibrios are just these one-off, you know, like, so what I can tell you, you know, I only talked about a couple of molecules, but every molecule, so we in my lab have found 4 molecules. Every molecule that we've found is a brand new molecule to biology, even though they're really simple, like that autoinducer 2,5, it's a brand new molecule to biology. The enzyme that makes it was just out of some genome that does brand new biochemistry, right? So if you think about the fact that we don't understand 50% of all the genomes of all the bacteria, it could be an incredible lexicon, right, of words, and that these are niche specific because they've had so much time, you know, to evolve them, right? And so, right, so I don't think every bacterium has hundreds of molecules. I think each bacterium has a few molecules, but they could have very different ones depending on where they are. And it could be the ones we have are the stupidest ones because we got the ones that have these huge readouts like, you know, bioluminescence. I mean, you have to be a moron to miss that, right? You know, it could be that the really interesting molecules, right, are actually the ones that just look like water to us right now, right? Yeah, I think that's going to be fun, right?
There are quite a few indwelling catheters and others that have the complications of infection because of the biofilm formation. So is it necessary that the molecule has to be soluble form?
Right, you're absolutely right. What's going to happen first, and what is happening first, it's going to be a long time before, I'm trying to make myself popular here by, you know, talking about this fantasy drug, but what is absolutely happening right now are going to be much more industrial things. So people are trying to hang these off of plastics for catheters. They're already in contact lenses, right? They want to put them in paints for cooling towers that get all gunked up. And so those industrial things where you don't put it down your throat and it doesn't get released, those are going to happen first, and they're happening. And so like the catheter guys, right? You know, if you're going to get a catheter, you're going to get a biofilm infection, right? And so there's a huge amount of work in that, right? So all the engineers are in it trying to link these, you know, onto these plastics, and I think that would be cool.
How is the toxicity of the chlorolactones?
Yeah, so you mean for the bacterium or for the host? For the host, I don't know, right? The worm, you know, so we can't, right? It's not a nice drug, right? And so, and the worm, you can't do that because the worm only lives 2 weeks anyway, right? And so we're trying to do that now in the mouse, right? But it's not a nice, I don't want to pretend that molecule it has to be refined, yeah. Mm-hmm.
I know a big concern in the medical community is antibiotic resistance. Is it possible that some synthesized or naturally occurring antibiotics can act as autoinducers?
So they do. So this is this business of like encoding lots of information. So we already know like some of these peptides that the gram-positives use. For me, it's an autoinducer. For you, it's an antimicrobial peptide. So some of these already are antibiotics. So the host, the producing bacterium has immunity, but if anybody's trying to listen into the conversation, they get killed, right? So for sure, antibiotics that we already know about might be autoinducers, and for sure some of the autoinducers are antibiotics, right? It just depends on who's getting it. Mm-hmm. It would be good to be able to do those double things with them, actually.
So everyone, there is a reception in the south lobby right immediately after this. Please let's thank our wonderful speaker again.